Yes, Team Euphrasia. It's a bit like a sort of rugby team. What I'm going to do is just take you through a bit of the history of Euphrasia taxonomy, so you know where we're coming from. It's a bit like that apocryphal Irishman. You see, how do you get to Dublin? Well, I wouldn't start from here, but this is where we are. And it all started with this chap. This is Richard von Wettstein, who was professor of botany at Prague and at Vienna eventually. And he wrote the first monograph, world monograph on Euphrasia. It's about an inch thick and is now, of course, way out of date, 1896. His friend, one of his friends was the MP for Bournemouth, who was Fred Townsend. I've got a soft spot for him. I was born in Bournemouth. And he had Wettstein's World Flora of Euphrasia translated, and he picked out the English or British plants, and he described 14 species and 6 hybrids. Oh, for the day. This is one of his. This is Euphrasia curta, which was described by a man called Elias Fries about 30 years earlier. And just hang on to that, because we'll come back to Euphrasia curta in a minute. It's a really good example of how Euphrasia taxonomy has worked. And things trundled along. People used Townsend's taxonomy until Herbert Pugsley, who was a fantastic botanist. This is him genuflecting to A.J. Wilmot. Obviously thought he was a fantastic bloke. And he published a British monograph in 1930 describing 20 species and 10 hybrids, some of the same ones as Fred Townsend, but he was very keen on publishing new ones. And one of the things he did was to start chopping up Euphrasia species into little pieces. And he did it with Euphrasia curta, which I showed you a picture of earlier on. This is one of the species he carved out of it. This is Euphrasia rotundifolia, and he carved out other ones, Euphrasia marshallii. So he began the process of splitting all this stuff up. We move on to the 1970s. Peter Yeo was a Cambridge graduate, did his PhD at Leicester on Euphrasia. And you can see that the numbers are creeping up, aren't they? He did some more splitting and split the Euphrasia into 23 species, but he was really, really keen on hybrids. He just couldn't explain all the variation which you know exists without involving lots of hybridization. And he also had a crack at Euphrasia curta, and I say it's a really good example of how this sort of stuff happens. He carved it up yet again and pulled this one out. This is Euphrasia ostenfeldii, Ostenfeld's Euphrasia. But then he got fed up and he left the rest. He refused to deal with them. So we have all these unnamed hairy Euphrasias, which we don't have a name for, which is a bit unfortunate. And nobody's done anything about it ever since 1978. Alan Silverside, who was the referee before me, carried on with the 23 species, but he was super hybrid man. And the account which he wrote for the Hybrid Flora in 2015 contains 81 hybrids and about half a dozen triple hybrids. Triple hybrids? Get a life, come on. And many of these have got tiny distributions. This is a triple hybrid between Micrantha scotica and Arctica. And you can see there are 2, maybe. So we then move on to when Fred and I authored the Euphrasia handbook. We managed to reduce the species by a little bit, but we had to retain all those hybrids. And you think, well, why on earth did we do that? I mean, that's a bit, a bit over the top. And the reason was really because we wanted to closely define the species. And the more closely you define the species, if you've got a very variable taxon, the bigger the gaps get. And so we had to maintain those hybrids to fill in all the gaps between the species. And this is a good example. We've got Euphrasia scotica on the left and Micrantha on the right. And in the middle is the hybrid mix between the two. And so the tighter we made the definitions of the two species, the more hybrids we had in between the two species. It was unfortunate. We didn't want that to happen, but it did. Over to Max.
So one of the reasons that Eyebrights are so problematic is because of their ecology and that they're very diverse. They're found in different places all over the British Isles. And they're also parasites. And we can attribute some of this taxonomic complexity to their parasitic condition. And within the Eyebrights, we've done a lot of work in the Alex's group on trying to understand what these plants actually grow on. And we found that they do grow on a large variety of plants across a huge taxonomic range. So they've got this wide host range, they've got this wide habitat range, and depending on the host that they have, they also seem to change their life history strategy. So they might grow faster and reproduce earlier, or grow slower and reproduce later, depending on their host. And if we look at the wild species in the wild, we find that the ecological boundaries between Euphrasia species seem to be quite blurred. So if we take a first look at morphology, we can see that how a Euphrasia plant looks is very dependent on the host that it's attached to. So we have here Euphrasia micrantha being grown in one of our many common garden experiments over the years. And you can see it's very stunted on one growth and very tall on the other. And actually, if we do an experiment where we take one species of Euphrasia, for example, if we compare the very left-hand box plots where they're grown without a host, they take a long time to flower. The flower size is somewhat a little bit smaller, and they're overall much more stunted. But if we grow them on a good host like clover, Trifolium repens, we find a very different looking plant. Secondly, if you grow different species of Euphrasia on the same plants, you can get convergence on some traits. This isn't always the case, but if we look at, for example, Euphrasia arctica and confusa, those first two columns of box plots there, actually across multiple traits, they're very similar. So that's the tetraploid condition. If we look at diploids, we also find the same thing. If we measure lots of different traits, we grow them on one host, and we try and separate these species out morphologically, we find it very difficult to resolve them. Finally, looking at wild data, so this was from the E4E project, which I believe was in 2015 or so, when people went out and they looked at individual plants of different species and the hosts that they co-occurred with. If you run an analysis on this, you find that individual species of Euphrasia don't tend to occupy a specific host type all to themselves. You get multiple species of hosts and multiple species of Euphrasia occupying similar niches. So it's unclear from this data if there are actually boundaries in host preferences between species in the current taxonomy. So there are a few ecological problems which we've sort of had to deal with. I'll hand over to Alex.
So good afternoon, everybody. Lovely to see you all. So I think this is my chance to summarize a decade of genetics work in 5 slides. So I hope you're all ready for it. So as Pete said in the introduction, Eyebrights are difficult, which I think you all know. So in terms of genetics, we have in Britain and Ireland, we have diploids and we have tetraploids, which makes it very challenging to analyze. Lots of recent species diversity, so species have evolved post-glacially. We have selfers, we have outcrossers, everything hybridizes, and as Max has just been saying, they're parasitic plants, so if you grow the same species with a different host, you get different morphology. So challenging, right? So it's a difficult but exciting group. And we're very aware that there is no simple solution to these taxonomic challenges. So what we're going to be presenting today is our idea for a consolidated, simplified, evidence-based taxonomy. So what does the genetic data tell us? So as Max mentioned, we had a project, I for Eyebright, 10 years ago with the BSBI, and many people here contributed. I asked you to send in your samples from around Britain and Ireland, and we got 276 populations. So thank you very much for your contributions. And a PhD student working with me, Ding, she analysed over 800 samples. So it's a huge effort across all of Britain and Ireland, and used 20,000 genetic markers. So a huge data set. And the clearest single result that came from this is that if you do a genetic analysis asking to separate the Eyebright into 2 genetic clusters, you get a very widespread group shown in purple, and then in yellow in the north you get Euphrasia micrantha and Scotica. So there's Euphrasia micrantha with some hybridizing Scotica come out as distinct. You can see that here if anyone's familiar with these type of structure analyses. Here you've got each individual, so the 800 individuals are rows, and you can see you've got a yellow cluster, which is Euphrasia micrantha and some Scotica, and then everything else is together. So micrantha is distinct, and I think many of you would recognize that in the field. You'd say that's an easy to identify species, hopefully. So the genetic data supports that. And then secondly, the ploidy groups are distinct. So diploids, we have tetraploids, and they're genetically distinct, as you'd probably expect. In Ding's analysis, when she asked the software structure separate 4 genetic clusters, she got all of the diploids together in red, Euphrasia micrantha, and then everything else together. We have analysed the genome size of hundreds of individuals, and you get a very clear separation between diploids and tetraploids. There probably are triploid hybrids somewhere, but we've never come across them. And I will save you the complexity here, but very talented postdoc Hannes Becker assembled the whole genomes of the diploids and the tetraploids and showed that our tetraploids are allotetraploids, so they're hybrids. Our British diploids are one parent, but they are genetically distinct. So the ploidy groups are distinct. But then I think we come to the really interesting stuff about what is not distinct, as many of you won't be surprised about. So many of the currently widespread recognized species are really not genetically distinct, and there's very little evidence to support them. So I think this for me was a really remarkable result. This was from Ding's data set, 800 samples, and we looked across all 20 species. And when we looked for signal of geography, we found a very strong pattern. When you look by clustering by species, you don't see anything. So for example, you get a genetic cluster in the southwest of England and Wales, which includes all of our common species. You get genetic cluster in England, Scotland, north of Scotland, etc. So you get this really clear genetic clustering by geography, but no signal of the species differences. And this is really striking. We did a field trip to Shetland with Jim and many others a long time ago and spent a lot of time analysing those samples. And all 10 tetraploid species sampled on Shetland cluster together and cannot be separated genetically. And just to highlight, for example, I'm sure many of you will have spent time maybe looking at Euphrasia arctica, Nemorosa, Confusa. They grade into each other. You can pull out extremes, so really they do intermix and they're not genetically distinct in any analysis. So a few other points before we come to taxonomy. In many other plant groups like Arabidopsis, Antirrhinum, the presence or absence of hairs has a genetic basis. So hairs are one key trait which we continue to use and we think is informative for the taxonomy. I wanted to highlight for anyone who thinks about genetics that there may be some genes underlying species differences, which we haven't found. So our understanding is still partial. We don't have all the answers, but we're basing it on what we know so far. And just to highlight that the selfing mating system, coupled with these local differences, mean there are numerous local, and I've tried to avoid using any sort of terminology here. You could call them local forms, varieties, ecotypes, variants. So we recognize there's local differences. We've probably all got our favorite local form or local variety. And we do need to have a way to recognize that and conserve them. But in many ways, genetically, none of these are really coming out as well-supported species, apart from a few hybrid species, which I'm sure we'll hear about shortly.
Hey, if anybody have read the account in Stace, the latest edition, this is what he says, sort of ringing in our ears, really, that Clive obviously had his finger right on the Euphrasia button, that a simpler, broader species limits would be the thing to do. So here we are. Now, a word of warning, you can't use this yet because we haven't published it. This doesn't count as publication, it counts as letting you know what's hopefully in the pipeline. So here are our species, reduced down from current 23. And I'm just going to go through them very quickly one by one. That's what used to be Euphrasia arctica on the right-hand side. And if we start off with, we haven't done any work really on Source Bergensis. It's only found in Ireland. And its next geographical location of the Austrian Alps. And we were talking over lunch, and I think the hot money is that it's probably not the same as the one in the Austrian Alps. I think we'd be very surprised, but we can't demonstrate that yet. Probably needs a new name in due course. And then, as you heard Alex say, the diploids split out very nicely genetically, and so they're all characterized by these long glandular hairs, which you can see. And so we've glued them all together as officinalis. They're currently subspecies of officinalis, but there doesn't seem to be any basis for that genetically. So we've glued all those together just as officinalis. We have maintained this one. This is we have maintained three species which we think are probably hybrid derivatives. Most two between diploids and tetraploids and one not. Very restricted geography. Vigursi is one, just found in the southwest of England, and Rivularis, which is an upland species, which is, again is quite distinct, found in three relatively small areas and one on the Isle of Mull, which may not be correct. You've already mentioned Curta. This is the species which we've resurrected back from Fred Townsend's day for all those hairy species. These are the E-glandular hairy plants, and also all those other unnamed things that our friend Peter Yeo couldn't be bothered to get round to. Well, hopefully now we will have a name for them. So we put all the glandular hairy plants into one, except for this lovely little thing. This is Euphrasia campbellii. It's only found on Lewis, and again, it's thought that it's probably a hybrid, stabilized hybrid derivative. More work to do on those, perhaps. Micrantha, again, you've already grasped, but we think this is very distinct. At the moment, it's split into two species. We think that the evidence suggests that it's better treated as one species with two varieties. There's variety var micrantha and var scotica, and so the big lump in the middle will just be micrantha ag or micrantha sensulata if you prefer. And then, and please don't think if it's a dustbin. Euphrasia doesn't belong in a dustbin. They're such beautiful plants, but that great big lump of plants that we can't actually separate genetically. All those lovely names and more that have been come up over the years and have been discarded. We've put those all under Nemorosa, which is the oldest name available for that particular group. Except for two, var frigida, which is an upland plant, and var tetraquatra, which is a very distinct plant, and it's coastal ecotype, rather as frigida as an upland ecotype. So we've separated those two out as varieties if people want to record them as that. Greg.
I get the exciting bit at the end, telling you what that all means, particularly as we have the GB Red List coming out for publication on November the 5th. So there's a sort of a little bit of a jumping the gun, because I happen to know what some of these things that we are about to perhaps soon lose would have been. So obviously the results of this study means that we have far fewer endemic species, and we all like our, as James has written a lovely book on this, we all like our endemic things which are special to us. But in these cases, we think there's very little genetic justification really for their continued recognition. We can think of them as sort of incipient species on their way. And I think part of the problem with Euphrasia is they've gone so far, and then we've just sort of mixed the landscape up and thrown all these things back together again, and that sort of re-homogenized things. So they've never really made it that final break into sort of speciesness. So as a potential, we've entirely sunk a whole range of these endemic species that we previously believed in. And there are species which we had an international responsibility for, in that probably more than 75% of their global occurrence were within Britain and Ireland. So things like Tetraquetra. Now we're fortunate in that although we'd recognised endemic species, actually none of them have ever been listed under any of the protection orders, which makes things a little bit easier, and perhaps gives some indication that people didn't really entirely believe in the worth of some of these things before anyway. But there are many threatened species, or things that were threatened, which of course have some conservation concern, which fall within this group of things that we are sinking or reorganizing. And so the taxonomic changes that we're proposing will impact what we do in terms of red listing and the conservation of these things. So we've lost our endangered species. Campbellii, as we've been hearing, is an intriguing thing. We're still not entirely sure how we best treat that, whether that is worthy of recognition at the specific level. And then there are these other things which are vulnerable and near threatened, which we are now no longer going to be believing in. And actually, it's mainly these things that we have no real sort of belief in that are the things which have had the higher threat listing. So they are the rare oddities, the local forms, which we are now sort of regarding as slightly sort of lower level. But I think it's fair to say that these things are still important. I think many of you will remember the one Scottish endemic fern, Ethereum flexily, which Heather McAfee and others in this building sort of worked on, and realised that it wasn't worthy as a taxonomic species, but was a distinct ecotype, which is endemic to an environment which is unique to this country. And it's sad that it loses its specific status, and then no one's interested in its conservation anymore. And that's really rather sad. And we don't want that to happen. These are important things which need to be conserved and considered, although they don't have this specific sort of tag any longer. So the impacts for you on the Scottish biodiversity list, only one of the 7 species which are listed we think would remain. That's Campbellii. We still need to work on that. But as Chris has said, that we have this sort of the Curta group, this hairy group, which had been so neglected in the past. And that's clearly where a lot of these really local, interesting, endemic derivatives are. So this is a group which does need some careful work, reconsideration, reassessment, because I'm sure that that will still come out as definitely threatened. Things need to be done. So we're not calling these species potentially any longer, but they are still important things. They're important. We've heard they're sort of fascinating study scientifically. You can look at active evolution here. You can look at co-evolution with their hosts. There are so many things which are very important to look at, and therefore they're worthy of protection. So it's still very much a process we're still arguing amongst ourselves as to how we best sort of do this sort of treatment, and we are open to lots of sort of comments and feedback from you, the people who have to pragmatically use the taxonomy that's created for recording purposes. But we just wanted to take this opportunity to sort of present where we're at and start to get sort of open the floor for feedback for what we're doing. So I hope I'm not sort of misrepresented anything there, and I think that's probably it for us, and I'm sure if there's any questions we can answer them either now or a little bit later.